Morning, everyone. Today, our group will be presenting about Epidermiphyton Floccosum. We are from Group 2. My name is Sarita, and my group member name is Hidna. So, Epidermiphyton Floccosum is a dermophyte fungus that plays a role in superficial fungal infections. Dermatophytosis, commonly known as ringworm, is an infection that affects skin, nails, and hair in both humans and animals. It is caused by a fungi that thrive on keratin, and these are grouped into three groups: Trichophyton, Mycosporum, and Epidermophyton. Epidermophyton floccosum is unique in that it primarily affects humans. Although it has a zoonotic potential, it is less commonly found in animals. So when diagnosing dermatophytosis, it is crucial to collect samples from the right area. The most suitable samples include hair, skin scrapings, and scabs or crusts, especially taken from an active edge of the lesion. This margin is where the fungus is most active and viable. If the infection involves the claws or nails, then nail scrapings or clippings should be collected. The two main methods used for sampling are scraping the skin with a sterile scalpel blade or plucking hair with sterile forceps. Before collecting samples, the area should be gently cleaned to remove surface debris, but alcohol should be avoided because this might kill the fungal elements. Collected material should be placed on a sterile, dry containers such as petri dishes, paper envelopes, or folded index cards to prevent contamination and preserve the viability of the fungal sample. Once we've collected the sample, the next step is direct microscopic examination. Typically using 10 to 20% potassium hydroxide solution, the process involves placing a small sample on the clean glass slide, adding a drop of potassium hydroxide, and covering it with a cover slip. After waiting 15 to 30 minutes or gently heating it, the slide is examined under a microscope. In positive cases, we can observe septate, branching hyphae, and sometimes arthroconidia, which are fragmented spores. In the case of Epidermophyton floccosum, we also look for macroconidia, which are smooth-walled, clubbed, or beaver tail-shaped and contain 2 to 5 internal septa. Notably, this species does not produce any microconidia, which helps differentiate it from other groups such as Trichophyton and Mycosporum. For a clearer observation, lactophenol cotton blue stain can be used, especially in cultured samples. The gold standard for confirming dermatophytosis in fungus culture, we typically use SDA, which is Sabouraud dextrose agar, or DTM, which is dermatophyte test medium, where a red color change indicates a positive result. The sample should be inoculated on the surface of the medium, but not very deep, to ensure adequate oxygen exposure. Cultures are incubated at 25 to 30 degrees Celsius up to 4 weeks. If Epidermophyton floccosum is present, the colony will appear yellowish green with a powdery or suede-like texture and a folded surface. The reverse side of the colony often shows a orange to brown coloration. Microscopically, we again confirm the presence of a club-shaped macroconidia with 2 to 5 septa and the absence of microconidia, which is a key characteristic of Epidermophyton floccosum. Now let me pass it on to Hitna to continue this presentation.
Okay, now I will move with the microscopic examination of native culture, also known as the tape prep. The microscopy of the natural preparation stained with lactophenol cotton blue revealed the presence of septate, hyaline, and branching hyphae, which is characteristics of dermatophyte fungi. Numerous macroconidia were seen, which were smooth-walled and distinctly club-shaped with 2 to 5 internal septa. The macroconidia were typically present singly or in small cluster. No microconidia were observed in the preparation. An important diagnostic feature about the Epidermophyton floccosum from the other two dermatophytes is that they characteristically have microconidia. The microscopic morphology observed is consistent with the identification of the E. floccosum, a dermatophyte causing superficial fungal infection of the skin, nail, and groin. This organism has the ability to infect keratinized tissue but not invade hair, which also fits its microscopic characteristics as observed. Here, for picture B, we can see the E. floccosum under the microscope. And I got this picture from Infeksi Jamur dan Parasite pada Kulit. It is a journal by the University of FK. On to the slide culture according to Riddle. So the purpose of this is to observe fungi in natural and undisturbed arrangement. This is suitable for filamentous fungi, which is the E. floccosum. For the procedure, first, the sterile slide placed on support in a Petri dish with moist filter paper. This is for humidity. The small sterile agar block is placed in the center. Fungus is inoculated at 4 corners of the agar block, and the cover slip placed gently over the agar. Incubation is 25 or 30 degrees Celsius for 3 to 5 days, and it's in a moist environment. This prevents it from drying. Upon observation, when the cover slip is removed and placed on a slide with lactophenol cotton blue stain, it shows clear views of spores, hyphae, conidiophores in natural orientation. And this is useful for accurate morphological identification. Next is the identification result. Identification of fungal isolates were carried out by microscopic examination of a native preparation and also verified by slide culture using the Riddle's method. Under microscopic examination using lactophenol cotton blue stain, the native amount revealed septate, hyaline, and branched hyphae and abundant smooth wall club-shaped microconidia with 2 to 5 septa. Microconidia were absent, which is a distinguished characteristic of the E. floccosum. To keep the natural orientation of fungal structures intact, a slide culture was prepared. The slide culture, after a few days of incubation, exhibited the same morphological features, the presence of clavate macroconidia in clusters and separate septate hyphae. The undisturbed position of reproductive structures in the slide culture also supported the identification. Based on the findings of both the indigenous microscopic and slide culture study, the isolate was identified as E. floccosum, a dermatophyte causing superficial skin and nail infection. In conclusion, the diagnostic of E. floccosum here was established by diagnostic process of direct microscopic examination, fungal culture, and slide culture using Riddle's method. The organism features septate hyaline hyphae, smooth, club-shaped microconidia with 2 to 5 septa, and an absence of microconidia. These are in line with features reported in the literature and confirm the presence of E. floccosum as the agent. This report emphasizes the importance of sampling techniques, appropriate culture condition, and morphological observation for accurate identification of dermatophytes. Although E. floccosum is a human pathogen, predominantly a human pathogen, its identification in veterinary sample highlights the relevance of considering zoonotic agents in veterinary dermatology. Continued surveillance and guided diagnostic practices are required to prevent the spread of dermatophytosis in settings where animals and humans interact. That's all for our presentation. Thank you.